Welcome. This is our first lecture and we'll be covering Chapter 1 in this lecture. And I just wanted to orient you, since this is our first lecture, I wanted to orient you to how I organize my lecture slides and give you some advice about how to use them to most efficiently in your studying and learning the material in the course this semester. Like subsequent chapters, I've organized the Chapter 1 lecture slides starting with the objectives of that I want you to learn from reading the chapter and listening to the lecture. I always start my lecture slides with the objectives, and then they're followed by some terms that you should learn, or if you're already familiar with them, great. Then I have the lecture material, and at the end, I have a slide that gives you some practice problems that you should look at from the text. Now, I would suggest that I've given you a PDF form of all of these slides, so there's no need to copy them down word for word. And I would suggest that you look at that PDF of the slides first before you go to read the chapter. Then you read the chapter and take notes, paying special attention to the objectives that I've listed on the first couple of slides. And then listen to the lecture, take additional notes to supplement what notes you've taken from the book. And then you can use those notes to study as well as to do the homework. So let's start with Chapter 1. Chapter 1, I'm going to give you an introduction to microbiology. I expect that you will understand the various members of the microbial world by the end of this lecture, as well as some of their important features and how they're linked to humans. And important features of the different members, how they are similar and related, as well as how they're different and how they're classified into the domains of life, and how some are actually not even considered living organisms, but are rather microbial agents. And then how these are related to us in a good way as far as our normal microbiota, as well as in the way that you will come into most contact with through your work in the healthcare field as pathogens, those germs that make us sick, so how they're linked to infectious disease. All right, so let's get started. I will let you look at the terms that you should be familiar with on your own. You should be able to get the definitions for these from the lecture. If not, you should find them in your book. Okay, so I just wanted to remind us all of what characterizes something as living. Because we are going to, being a microbiology course, we are going to study biology, the study of life. And so what is it that makes or defines something as living and something as non-living? And this will become important to understand in understanding key differences between members of the microbial world. So the 7 common traits are that all life is composed of cells. There are levels of organization. Most commonly in microbiology, these are very small single-celled organisms that we're going to be studying. But whether an organism is unicellular or multicellular, as we are, there are different levels of organization, and that anatomy and the physiology of the organism is what we'll be studying as part of our course this semester. All living things use energy. That energy helps them to drive their metabolism, and it helps them to grow and reproduce. And key molecules within These organisms enable them to respond to stimuli, so they're key communication pathways that allow for the response to stimuli, as well as through reproduction or other means, living things can adapt to their environment. And that's true of microorganisms as well as other macroorganisms that you've already studied. For example, the human body. So remember that you've already studied a lot about the human body and these traits in the human body. But when we come to study microorganisms, these are really very tiny, invisible life. And so for a long time, no one knew that microorganisms even existed. So biology was a field devoid of the study of these tiny organisms. But we understand now from so far what we've learned about these tiny organisms is that they probably were the first forms of life on Earth, and that today they are essential to our survival, not just those microorganisms that are directly associated with our body, but those that are in the environment, and they exist in all ecosystems, all biomes within our Earth. And so it's essential, these microorganisms serve roles within our environment of recycling essential nutrients, organic molecules, organic elements that are necessary for our bodies to function, our metabolism to occur, and for us to be able to have enough oxygen to breathe, etc. And so over time, though, just like we talk about human evolution, microbes have changed. And their relationships with other organisms, multicellular organisms, have changed as well. And so we'll look a little bit about how our relationship with these tiny invisible organisms, what we know about it so far. So I use two terms, microbes and microorganisms. They're usually used interchangeably, but I'll come to a key difference in a moment. But as far as microbes and humans, there's good news and bad news. Good news is that we have about 10 times, or probably more than 10 times, as many microbial cells associated with our body, either in our body or on our skin, than we do actually have human cells. So just by the sheer number, 10 times more microbial cells than our own human cells, we can infer that these microbes are really key to our survival and our development. And through the Human Microbiome Project, we're really learning more about this relationship of these good microbes and the microbiome, the environment of our body, and how it serves as a habitat for these microorganisms. In addition, we rely on microbes for food production, for preserving food, for treating infection, for mining precious minerals, and for bioengineering, because many of them serve as biotechnology tools, so developing new medicines using microorganisms or their products. The vast majority of microorganisms that are on our Earth are harmless to us, but of course, even though it's a small number of microorganisms and microbes that affect us negatively in causing infectious disease, it is a significant and critical proportion of these microorganisms. And so a lot of the semester we'll be talking about some examples of microbes and how they cause disease, and why they are really the leading cause of death for humans worldwide. Okay. So what is a microbe? Well, as I mentioned, a microbe is a microorganism, a very tiny, invisible form of life. And the simple definition is that it is simply, a microbe is simply a tiny, invisible, to the naked eye form of life. But it gets more complicated when you start to study the variety of microbes. So, for example, some microorganisms can actually be seen with the naked eye, so they aren't invisible. And others are able to grow multicellularly, and so again, we can see them without any tools to aid in making them bigger so that we can see them. In fact, many microbes that are unicellular form communities in their growth called biofilms. So for example, bacteria. And these communities can be seen by the naked eye. So they're macroscopic. In addition, some microbes are not organisms. They're not considered living. They lack a cellular structure. They may even lack genetic material in the case of prions. And so these organisms kind of fall out of that very simple definition of a microbe as a microscopic organism. But for our purposes, we'll stick with this simple definition and with the assumption that you understand these key caveats. So let's look at the members of the microbial world and a little bit about how they are similar and how they're classified and how those differences are used to classify them. So all members of the microbial world are microbes, but only some of them are organisms, as I alluded to when we were talking on the last slide. So if we're just looking at the microorganisms, they can all be classified into those three domains of life that you learned in general biology: bacteria, archaea, and eukarya. So these three domains can be divided into two groups based on their cell structure. Prokaryotes, the prokaryotic cell structure, where it's unicellular, has no internal compartments, no nucleus, are the cell structure of both those microorganisms in the domain bacteria, as well as microorganisms in the domain archaea. But microorganisms in the domain eukarya have a eukaryotic cell structure. It's more complex, has internal compartments that are bound by a lipid bilayer, a membrane, a biological membrane. And so all of these members of the microbial world, algae, protozoa, fungi, and helminths, are all part of this domain of eukarya. Now there's a dashed line to helminths because unlike algae and protozoa and some fungi that are unicellular, helminths are always multicellular and can always be seen with the naked eye in their adult form. However, they do have microscopic forms of their life cycle, like the egg cycle. So they are, and they also cause infectious diseases, so they are grouped and included in these members of the microbial world. On the other hand, there are some microbes that are non-living. They're infectious agents or microbial agents, and those include viruses, which you're familiar with, viroids, which are a simpler form of virus that only infect plants, and prions, which have no genetic material but do cause infectious neurological diseases like spongiform encephalitis. All right, so Just an aside for a moment on nomenclature, because you'll see the names of organisms within your text, and I just want you to be familiar with how microorganisms are named. And that's following the Linnaeus's two or binomial nomenclature, where the first name is the genus name, and the second name is the species epithet, or the species name, a specific name for a subset of organisms within this genus. And the names are always written with the genus name capitalized and the species name lowercase, and they're always in italics or underlined. We can abbreviate the genus name, but we never abbreviate the species name. So Escherichia coli would be E. coli, more familiar. In looking at how these microbes are related and how they differ, we first need to step back to our basic understanding of biology. And I just want to refresh your memory about the genetic basis of life. So remember that DNA, deoxyribonucleic acid, is this self-replicating material that's found in all living things that carries the information that defines that organism. Okay, so all the information is contained in what we call a genome. It's unique to that species, and it's found in all cells within that species or within that multicellular organism. Genes are a portion of that genome, a portion of that DNA that carries a specific trait of an organism. So we can actually study DNA, its sequence, the sequence of bases here in the middle of this double helix, the gray and the blue and the red here in the center of the double helix are the bases, and there's a certain sequence to them. The adenines, guanines, cytosines, and thymines, that if it's very similar, then that means that organism is more closely related than if the pattern of these nucleotides is very different. And the way that we determine that is by sequencing the DNA, or determining, or kind of reading that code in order by doing DNA sequencing. So the basis of all disease boils down to what genes those microorganisms or microbial agents have. And really the difference between those microorganisms that are harmful to us, that are pathogens, and those that are benign or beneficial to us is based in their genetic makeup. So as I mentioned, there are these two major groups based on the anatomy or the structure of the cells that make up these organisms. And so you should know the difference. You should have already learned this in general biology. Prokaryotes have no internal compartments, no internal lipid bilayers. They simply have one lipid bilayer around the entire cell. That's the cell membrane. So they don't have any nucleus or any organelles. And this includes all of the microorganisms in the domain bacteria and archaea. Eukaryotes, on the other hand, are more complex. They do have internal compartments. They have a nucleus that's bound by a double lipid bilayer called the nuclear envelope. And they have internal membrane bound organelles such as the endoplasmic reticulum, Golgi, lysosomes, vacuoles, endosomes, etc. So review these. I'll expect you to know those differences between prokaryotes and eukaryotes. And these include microorganisms such as fungi, protozoa, algae, and helminths. So just quickly to introduce you to the various members and help you to at least initially know some key distinctions between them. The bacteria and the archaea are very similar. They're both prokaryotic. They're both around the same size, although this size, so a typical bacterium is about 1 to 2 microns, but it can be, some bacteria are actually so big that you can see them without the aid of a microscope, so they can be quite big. So there's always exceptions to the rule, but on average, they're about 1 to 2 microns, micrometers. And this symbol here that looks like a U means micro, and then the M is the meter. So micrometers are microns in size. And you should review your, these are metric measurements, so review your metric measurements if you're a bit rusty on that. We'll be only using metric measurements in this course. Bacteria are unicellular. They only have one cell, but they do grow as groups of cells, communities that we call biofilms, or they can grow individually, or they can form filaments. And bacteria are found virtually everywhere. Everywhere we've looked, we've found in the environment, we've found bacteria from the geysers of Yellowstone Park to the forests to our own body, etc. Archaea, on the other hand, they're genetically distinct and they have some key differences in their cell wall composition, so anatomically they're distinct. And they're called extremophiles because they're not found everywhere in any environment, but instead in very hot or very dry or very acidic or very salty environments. So hence their nickname extremophiles. So down at the bottom of the ocean under incredible pressure, archaea can live in deep sea vents that spew out very hot gases from the core of the earth. Another key difference is that bacteria, a small portion of them, are pathogenic, whereas we have no known record of any archaea being pathogenic. So they're non-pathogenic as far as we know. So moving on to the eukaryotic members of the microbial world, protozoa and algae are protists. They're unicellular. Algae can be also multicellular, but we'll be focusing on unicellular. And in the case of protozoa, they're defined by their motility, so they're grouped and classified by their motility. And they can be either living free or relying on a host to survive. And in that case, they're parasitic. So they can be pathogens of humans or other animals. Algae, on the other hand, they're mostly in aquatic environments, although some are found on the land. And they're unique because, or different, distinct from protozoa because they are capable of carrying out photosynthesis. They contain chloroplasts, which help to capture that light energy and convert it into a useful form of ATP for the algae to grow. And as such, they serve kind of as that basic food source. There are even protozoa will even feed on bacteria and algae as part of their diet. Fungi, also eukaryotic, however, they are not motile. They rely on spores that passively travel through the air in order to move from place to place, from a nutrient-rich environment to the next nutrient-rich environment. They grow by absorbing nutrients across their plasma membrane, but they also have a cell wall, which is important, as do algae. However, protozoa lack a cell wall, and that's also a distinction between them and other microorganisms of the eukaryotic group. And then just an aside about cell walls, fungi have a distinct cell wall composition that's not like bacteria. It's mostly chitin or other glycans, and also distinct from algae, which have a cell wall made similar to plants, made of cellulose. So the vast majority of fungi are saprophytic. That means they're growing on dead and decaying material, and they're important recyclers in our environment. But some of them can cause disease in humans. They rarely cause a primary infection, meaning they rarely cause disease in healthy individuals, but they often cause disease in immunocompromised or individuals with a weak immune system. So they're opportunistic pathogens, that's what we call them. Viruses, on the other hand, are the example of the non-cellular microbial agents. And they are composed of, so they don't have any organelles that would enable them to reproduce independently, and so they lack that cell structure. However, they do have genetic material. They, unlike living organisms, which have both DNA and RNA, so two forms of nucleic acids, viruses only have one or the other. But the genetic material that they do have carries genes that are necessary for producing another complete infectious virus particle. And so viruses are required to use a host cell because they're incapable of reproducing outside of a host cell. It is necessary that they take over the metabolism of a host cell in order to reproduce and make more virus. So they have some qualities of living organisms, but not all of the qualities. They're important human pathogens, as well as now they're often used as biotechnology tools in treating disease, for example, cancers, in delivering the molecules or the agents that are used as anti-cancer agents directly to that tumor. Other non-living members of the microbial world include viroids, which are also acellular or non-cellular. They don't have DNA, but they do have genetic material in the form of RNA, which carries genes that are necessary for them to generate more virus. And they don't cause disease in humans, but they do cause plant disease. And then finally, prions, which really, within my lifetime, set molecular biology on its head by the surprising finding that it was indeed an infectious agent that contained no genetic material at all. So no DNA, no RNA. Normally, the protein is found in our neural tissue, but if you somehow acquire the misfolded protein, either by ingesting contaminated meat, such as mad cow disease, would transfer that misfolded protein, or by genetically inherited form, as far as Creutzfeldt-Jakob syndrome is concerned, that misfolded protein can then cause the normal protein to become the disease form. And hence, over time, you get this neurodegenerative disease. So we know that microbes exist now because we can see them. But prior to that time, it was diseases were known, but there was no link between diseases and their causative agent. So really, the advent of microscopes were the first introduction to the microbial world. And Robert Hooke in 1665 was the first to build a compound microscope. And he looked at various tissues, plants and animal tissues, and he noticed something about them that they had in common. Here in this drawing that he made that was published, so really the first published record of the organization of tissue, this is from a cork plant. And you could see these kind of repeating, regularly repeating boxes that he noticed in other organisms, especially plants, especially obvious in plants. And these he called cells because they reminded him of the small compartments that monks lived in monasteries. And so it was really his observations through this compound microscope, meaning it has two lenses and so can give greater magnification than previous lenses. It's really through his observations that we began to understand that cells are that fundamental unit of life. Now, he didn't look at microorganisms, but another individual who was actually not a scientist or a doctor, but he was a drapery merchant, and he needed to use the lenses to look at the details of the threads. But he was particularly adept at grinding the lenses and got, even just with a single lens, much more powerful magnification than Hooke did with his compound microscope. And so he was able to look through these lenses, and he was just curious about things in general, and so he would look at various things, including a drop of pond water, and he published these drawings of what he saw in the pond water, these tiny rod-shaped or circular organisms, or even wavy organisms that moved from point to point. And these he called animalcules, but really his was the first written record of microorganisms. So at this time, people did not really understand, a natural question that would arise from these tiny organisms is, where did they come from? And people were always questioning, where does life come from? And with multicellular organisms, humans, it was clear that humans would mate and then they would have children, and so humans came from humans. But what about these tiny newly discovered organisms? And so for many biologists, they believe that they just kind of magically appeared, that many organisms were able to spontaneously generate. And it wasn't until some really extensive experiments over almost a century of time that we understand really the biogenesis is responsible, so pre-existing organisms is responsible. So I just wanted to go over some of these experiments that are key to our understanding of how microorganisms arise. So in Italian, Spallanzani used a kind of ground up meat broth and observed that if he left a flask of this meat broth open to the air, like a beaker, it would become cloudy and full of life. But if he boiled the broth and then left it covered, he would not find any life. So he sealed, if he stoppered it or sealed it somehow so that no air could get in it, then there was no life. It remained clear. So this was the first indication that it wasn't just that nutritious broth that was like spontaneously giving rise to this new form of life. But his detractors said, you know, you really have blocked the entry of oxygen, and so that's why the life can't grow, because they need oxygen. And so later, Louis Pasteur, a French scientist who was fascinated with the spoilage of food and the making of wine, and he developed a kind of flask that was still open. It's called an S-neck or a swan neck flask, and you can see a picture of it here. It was curved, so it was still open to the air, but that any kind of dust or sediment in the air would be incapable of reaching the nutritious broth inside the flask. And after he boiled the broth and let it sit, he found that the broth remained clear. So this was really a critical turning point that debunked spontaneous generation, that these broths themselves could not spontaneously give rise to life. Many people tried to repeat his experiment and found that the results were inconsistent. And John Tyndall was one of those, actually an Irish physician and scientist that found that sometimes he would boil the broth and it would still give rise to a cloudy, growing media full of microorganisms, and other times it would remain clear. So he eventually determined that there was some more resistant, heat-resistant form of life that we now know are endospores produced by bacteria under very stressful conditions. And so now, in order to truly sterilize materials, whether they're liquid or tools for instruments for surgery or anything that needs to be truly free of microbes, we use an autoclave in which rapidly boils the liquid, if you have a liquid, or heats up the material, but also has high pressure to destroy those endospores, so that really, truly, the material is free of microorganisms. And in the lab, all of our lab materials are autoclaved, all of them that are not sensitive to heat. So here with this series of experiments, here shown in more detail in this figure, where the broth on the swan neck flask is boiled, it's allowed to cool, and then for years, even there's still a flask in the Pasteur Institute in France that you can go and visit today that's still, to this day, more than 100 years after the experiment was done, is clear. But he found if he tipped the flask and allowed the broth to come into this swan neck, or if he broke off the S neck, then just a matter of hours or days later, he would get a cloudy broth that was full of life. And so this was really the beginning of the understanding that all living things arise from pre-existing life, the theory of biogenesis. And later that was extended to all cells arise from pre-existing cells by a Russian investigator, Rudolf Virchow. Okay, so I wanted to finally move on to this final part of Chapter 1, which is really an overview of the course. And a lot of what we're going to do in the course is to talk about how microorganisms are linked to disease, even though the majority of microorganisms that we know of are harmless to us as humans. Obviously, for clear reasons, we are going to focus on that small portion of microbes that are associated with human disease. So what is it? The big question that we're going to address is, what is it that make these microorganisms that cause disease in humans different from all of those other microorganisms that are essential to our ecosystem, but that are harmless to us, right? So one of the first observations, even before we knew what microbes were, we understood what disease was. We understood that disease could cause the human organism to go into a state of ill health, and that the state was altered from that healthy status of the organism. And so it wasn't really until the first observations of these tiny invisible organisms that scientists and physicians started to make a link between and get some idea that there was a link between these tiny invisible forms of life and the health status of humans. And so there are a few really important contributors to this idea. One was a Muslim physician and philosopher, Ibn Sina, who noted that specific diseases specific diseases are caused by specific kinds of microbes. So he made that link between microbes and disease, and that not every microbe would cause the same disease, that different microbes were associated with different symptoms. Also, Florence Nightingale, a really influential figure in the field of nursing, first to write a nursing textbook and really to establish a curriculum for training nurses, and also the first to really record the significance of death due to disease, due to infectious disease. And here's a figure from one of her publications where she basically graphed out and did the statistics to show that infectious disease was a major contributor to death during wartime from 1855 to 1856. And you can see that by the blue wedges, which are representing the infectious diseases, being much larger than the pale red wedges, which are death due to battle wounds, which also could be as a result of sepsis or the presence of an systemic infection with microbes, and then other unknown causes in the black wedges. So you can see during this period of time from April all the way to November and December, the largest wedge for most of these months was the mortality or the deaths due to infectious diseases. And then finally, later, Robert Koch developed steps to actually prove this relationship that Ibn Sina had originally made noticed between that link between microbes and specific infectious diseases. And so he developed a certain sequence of steps that you could do in order to really demonstrate that link. And I'll come to that in a minute. But really these statistics that Florence Nightingale began collecting and then became the foundation for what we now call epidemiology, or the study of public health. And these statistics are really key in highlighting what would otherwise go unnoticed as far as the trends in infectious diseases. So here are a couple of graphs that are typical of recordings of for infectious diseases, or made by epidemiologists in their practice. So the first one on the top here with the blue bars is the number of hospitalizations. So you can see the weeks of the year are on the x-axis, and you can see that from weeks, say, about 42 to 45, there's a peak in hospitalizations. And then down at the bottom here in these gold peaks, gold bars show the number of deaths at that hospital, and you can see that the hospital deaths follow quickly after that peak in hospitalizations. There's a subsequent peak in deaths. So these kinds of observations are important for improving public health, improving practice in hospitals to avoid and prevent not only deaths, but also the spread of disease. And two organizations that are really major contributors to this worldwide are the Centers for Disease Control and Prevention here in Atlanta in the U.S., also known as the CDC, and the World Health Organization, also known as the WHO, which is more involved in worldwide, not just national diseases, but also diseases worldwide. And really in the world we live on, with such frequent global travel, most diseases are a global concern if they're highly infectious. So I'm just going to take a moment to go over this interesting case that's outlined in Chapter 1. A farm worker had the job of burying cows that had died of anthrax. Anthrax is an infectious disease that kills domesticated animals like cattle and sheep, and it's caused by a bacterium called Bacillus anthracis. And although he took care to wear gloves when handling the carcasses of these animals, he did come down with an infection shortly after performing this job. So these lumps that he found on his cheek progressed and got worse and opened, and so he went to the doctor and he wanted to find out what was going on. It didn't look normal, and so the doctor found that there was a very firm nodule of tissue that had kind of inflamed reddish purple ring, and then there was black over the dead tissue that was in the center of the ring. He prescribed antibiotics, which is standard for treating a skin infection form of anthrax. And the patient showed improvement upon this treatment. And in testing the patient just to confirm that his diagnosis was correct, the patient's serum, or that portion of blood that doesn't carry any of the blood cells, was tested for antibodies that were specific for the microbe or the bacteria that cause anthrax, the Bacillus anthracis. And indeed, it revealed the presence of antibodies specific for just that bacteria present in his serum. And so that was a way that the laboratory test could confirm the doctor's initial diagnosis. So the interesting thing about the Bacillus species, not just Bacillus anthracis, but also other species, is that they are endospore-forming bacteria. And we'll learn more about this important dormant or resting form of bacteria that differs from the actively growing stick-like bacteria in this scanning EM. So the spores are in the red here, or the pink, and the normally growing, multiplying bacteria are in this kind of yellow-gold color. And we'll talk more about that when we talk about the structure of bacteria and the various forms of bacteria. So the interesting thing about anthrax is that it happened also to be the organism that Robert Koch was studying when he developed the Koch's postulates. And what he did was he developed a way of actually growing the microorganism, the bacteria, in a lab setting. And he discovered a way to grow just that one type of bacteria and no others. So which is called a pure culture. It's only growth of a single type of microorganism in this kind of nutrient-rich media, which can be either in a solid form or a liquid form. And we're going to learn about and use these kinds of techniques in lab this semester. So the steps that he established to link a specific pathogen with a specific disease are these 4 steps. First, you had to find the microbe in all cases of the disease, but not in healthy individuals. Second, you had to isolate the microbe and grow it in pure culture. Third, you had to test the microbe's ability to cause disease by introducing it into a host animal or into a healthy human, and waiting to see if the same signs and symptoms occurred. And then finally, you had to, once again, isolate that same microbe from the test animal or test host. So these 4 steps are important criteria that have enabled us to identify the cause of many, many infectious diseases. However, they don't always work for every pathogen. So for example, Mycobacterium tuberculosis, which is the causative agent, it's a bacteria that causes tuberculosis in humans, actually only causes symptoms in about 10% of the people who have been exposed or infected with this bacteria. So step 1 was not fulfilled in that case. In the case of HIV, it's often at very low levels and very hard to detect in the early stages before it progresses to AIDS. And also the host, there's not an effective or a good animal host for experimenting, and of course experimenting with humans would be unethical. So there's a limitation in how far you can go with these 4 postulates in the cases of some infectious diseases. One story that I want to share with you, though, is about Barry Marshall, an Australian physician who was so convinced because of the correlation that he saw with many, almost all of his patients having a bacterial infection with Helicobacter pylori when they came in and complained of symptoms of ulcers or ulcerative colitis, so an inflammation of the colon in the form of ulcers. And when he tested these patients, he found that they had Helicobacter pylori in their stomach, and it was colonizing the stomach. And so he boldly stepped up, and he and some of his lab co-workers decided to test the Koch's postulates on themselves, steps 3 and step 4. And they actually drank a pure culture of Helicobacter pylori, infected themselves, showed that they got the same symptoms as the ulcer patients, and then cured themselves with an antibiotic. And really it wasn't until this bold move that the rest of the medical community, who was convinced it was simply lifestyle changes such as high stress that would contribute to ulcers, But so until then, he had Barry Marshall had a very hard time convincing the rest of the medical community that this was not simply a lifestyle symptom, it was actually an infectious disease. And so now routine treatment of the symptoms of ulcers is to test for Helicobacter pylori and then to treat with antibiotics. So one other topic that we'll be covering in our study of microbes and their association with humans and their link to diseases is how we control these microbes. So I've already mentioned about antibiotics, and we're all familiar with how antibiotics can control or cure a person of an infection. They're also early on, when even really before microbes were observed, there were some physicians that noticed that there was a correlation between the patient death and the presence of microbes. And so one of those physicians was Ignaz Semmelweis, and he found that there was a very high mortality rate in the maternity ward of his hospital, but that it was often correlated with physicians coming from the surgical suite, surgery on another patient, and without washing their hands, going to aid in a delivery. And so he ordered doctors to wash their hands in a chemical chlorine that served as an antiseptic. And by that I mean a chemical that kills microbes. And he found that the mortality rate fell by 80% after that. Still, doctors were set in their way and they refused to accept his findings. But of course, now we know that hospitals routinely have hand washing protocols in place to prevent the spread of infection from patient to patient. Another important historical figure in this effort to control pathogens was Joseph Lister. And he noted that again, he was a surgeon, and he worked, in this case, with a lot of patients that were undergoing amputations, and he noticed that about 50% of his patients died after the operation of a systemic infection called sepsis. So he began to use antiseptic agents directly on the skin of the amputee, as well as to pre-treat the surgical instruments in between using them from one patient and another. And he found a dramatic drop in the rate of death after amputation as a result of these simple steps. And so today it's really important for the surgical suite to actually be what we call fully aseptic. It's almost completely, we can't really say completely free of microbes, but it's almost completely free of microbes. Certainly the surgical instruments are completely free of microbes. They're sterile, and extra time is taken for the washing of hands of those involved in the surgery. Surgical gloves are worn when when undergoing the surgery, and the patient's skin, which is now actually one of the sources of infection post-surgery, is also treated with antiseptics that can provide a somewhat aseptic environment even on the skin of the individual undergoing surgery. So as I mentioned, another way to control pathogen is the use of antibiotics. So how did we come about discovering antibiotics? And that was really a key observation made actually just serendipitously, just by accident, when Alexander Fleming left a plate of the bacteria that he was studying out over a period of several days when he was out of the lab. And he came back and was going to throw out the plates since they were quite old by that time. He normally only grew the plates overnight, or at the most over 2 nights. Except for that he noticed that there was something else growing on one of the plates, and that something else had prevented the growth of the bacteria in that area, had actually killed off the bacteria that had previously been growing in that area of the plate. And so you can see over here this white dot represents that mold that he found growing on the plate, and the smaller gray dots and areas of growth that you see on the dark background, which is the nutrient-rich media that the bacteria growing on that nutrient-rich media. So you can see right around that large white dot of mold, you don't see these smaller dots of bacteria growing. So that was really an important observation, but he was actually unable to figure out how to purify whatever it was that the mold was producing that was toxic to the bacteria. And it was instead a couple of other biochemists that were able to purify it, one of which was Ernst Ch1. And so once they purified it, they were able to test it in a patient with a staphylococcal infection. And Staphylococcus was the various species, Staphylococcus aureus being one species that was studied by Alexander Fleming, causes a skin infection in humans. And so this antibiotic was tested in an individual, actually a British policeman that was sick with Staphylococcus. And he made a remarkable recovery before actually going back into, before the bacteria actually ended up regrowing and reinfecting him and actually leading to his death because they had run out of the antibiotics. So, but that was really the first demonstration that antibiotics could possibly cure individuals of infection. And we now know that antibiotics are used routinely to treat bacterial infections, and really it has been revolutionary because most of what threatened the health of individuals prior to antibiotics were common bacterial infections that would result sometimes in it being necessary to remove that part of the body that was infected in order to prevent a systemic infection or sepsis, which is highly fatal. Okay, so I'm going to leave you with that and just remind you that there's some practice problems in the book. You should be doing the thought questions throughout the chapter. The answers are in the back of each chapter. And then I have highlighted a few of the review questions and the clinical questions at the end of the chapter that I think you should focus on. And again, the answers you can find in the back glossary of the book.